Hi there, my name is Marissa Estrada. This is Lab 8 results video. It's December 6, 2020. This is Biology 225 of the Fall 2 2020 semester. So to start off, I washed my hands, I donned my PPE, I also sanitized my workstation with the 10% bleach solution. I have my lab printed off, and I'm also recording on my laptop, and I have my phone here to take photos as needed. I have my results here. It's been, I'd say 16 hours-ish, maybe less than 24 definitely, but around between 16 and 24. I don't want to do the math right now. Since I did this experiment, I was supposed to do it at 9 a.m., but majority of these had not turned. I was quite cold in my house this morning, so I'm thinking that that was the reason why incubation took a little bit longer. But I noticed on here last time that it says not to incubate longer than 24 hours, as the results may be inaccurate. And it hasn't. Yesterday I did these at about 9:30 p.m. and it is about, it says 7 right now, so like 7:10. Okay, let's see. I looked over my results and I wrote down little notes here too of what I was thinking as I saw them, so that way I didn't forget to say anything. So I'm gonna read through the experiment and get going. So observe the phenol red broths after 12-hour incubation for color change. If no changes have occurred, incubate for an additional 6 hours. Do not incubate the phenol red broths for longer than 24, as extended incubation times will result in inaccurate results. Okay, so we're within our time. Wipe down your work area with 10% bleach solution. That's completed. Wash your hands thoroughly with soap and warm water. Completed. Put on your goggles, a new pair of gloves, face mask, and apron. That's completed. Gather the six incubated phenol red broths. They're right here. Observe each broth, noting the color of the solution and the presence of gas bubbles in the Durham tubes. So here we have our number 1, the S-cerevisiae glucose, and I'm noticing bubbles at the top, and I'm also seeing the color is definitely changed. So what I put on my thing is it definitely is a fermentator and with gas production, and it's yellow, and I don't see any turbidity in this. It's pretty clear. I'm not sure how well you can see it. Okay. Next, I have my S-epi glucose, and this one is it's a little different in color, maybe just a little bit more pink than red. There's no turbidity, so I'm going to say this is a non-fermenter. Okay. And then I have my S-cerevisiae fructose. S-cerevisiae fructose. And this obviously changed color. It's now yellow, and it's slightly turbid. I'm not sure if you could see. It kind of all looks the same. I'm trying to see if there's an angle I could show you that. And you probably be able to see it in the light better, but there's definitely some turbidity here. Let's see, where was I? So that is these are results of gas and acid production of fermentation. Okay, so this is my S-epi. See that? S-epi fructose. So this is the color slightly changed. It's more pink than red. Let's put it in the light. Pink than red. Let's see. No turbidity. You could see it's totally clear. I hope you can see that. Obviously you'll get the photos into the lab or in the HOL website as well. So if you can't see, I just really want you to be able to see the colors and the labels. That's the big thing, so that way, I don't know, just making sure that everything's on the up and up. Okay, so no turbidity, so this is a non-fermenter. This is my S-cerevisiae mannitol. So this is kind of funny. It's not the same color as the S. cerevisiae fructose, but it definitely changed colors, more of a yellow. Maybe it needed a little bit more time, but it's gonna hit the 24-hour mark, and we need to make this video. So it's slightly turbid, just a tiny bit. And there's also a gas bubble within the durum tube. So I am going to say that this is a fermenter with gas and acid production. So this last one is S-epimannitol. S-epimannitol. And this one, the color changed slightly. It's more pink than red. And what else did I write on here? There's no turbidity, and there's no gas production. So I would say it's a non-fermenter. Okay. So I'll be recording all these results into data table 1 after I complete this video. Categorize each result as fermenter with acid production only, fermenter with acid and gas production, or non-fermenter. So something broke in my house. I think that it was my cat or something. Okay, sorry, that threw me off. It scared me. Let's see, where was I? Ooh, that startled me. I don't know why that threw me off so bad. Okay, so this is my S. cerevisiae glucose, and I would say that this is a fermenter with gas production. I kind of already went through these. I don't want to make the video longer by I told you which one each was. Okay, let's record your results in data table 1. Take photos of your results and upload from 1 through 6. Okay. Soak reusable equipment such as test tube clamps and racks in 10% bleach solution for 2 hours after contact with active cultures. Reusable materials should be rinsed with tap water and allowed to dry before returning to kit. Okay. So I think we're good. So what I'm going to do, I don't think that you need to watch me, is I'm going to be taking photos of each one and then uploading them to my A2L website. After that, I'm going to be disposing of these. How does it say? Soak all equipment. Dispose the liquid cultures by pouring BIOL contents into pure bleach solution. Soak the BIOLs in a pure bleach solution for 2 hours before wrapping them in paper towels, sealing in plastic bag, and placing in garbage. Secure all disposed items out of reach of children and pets. Okay, so I'll be dumping all these in pure bleach and soaking the test tubes in pure bleach and then tossing them after 2 hours. Okay, I'm trying to think if there was anything that I could work on with this lab. Oh, one thing is to make sure my house, because it sounds kind of funny, but what I do is I incubate my microbes in my oven, and I have a little sign on my oven that says not in use, so that way my boyfriend doesn't try using it, but he knows that they're in there because it's the most insulated part of my house. My house is kind of cold. We run the heater, but not very often because we kind of like it cold. I don't know. It's just our preference. But we need it to be warm. We need a place for my microbes to grow for this class. So the oven works wonderfully, and we just take them out and put them in a cabinet until we're done with the oven, and then we put them back. So I think that what caused this is just our house was too cold, and the oven just was not warm enough even in the oven for it to be warmer. So I think that next time when I incubate more incubate more microbes, is I will be ensuring that when I'm growing the microbes, the days I'm growing the microbes, my house stays within that 21 degree to 25 degrees Celsius range. So that way I don't have to worry about, oh, my computer's falling asleep. So that way I don't have to worry about the microbe's not growing or taking too long. Okay. I think that's good. If you have any questions, please feel free to email me. Have a wonderful night.